So we are looking into a very fascinating topic today, one that is not only something which needs to be learnt, something that almost challenges our knowledge, basic knowledge of molecular biology and gene regulation, but also because it is of supreme importance in our lives, and it is the basis why more than 80%, more than almost 90% of people today infected with COVID are defeating the disease, and the basis to our vaccinations, and that is the generation of antibody diversity. You see, we all know that we get the one gene, the traditional knowledge is one gene, we get one mRNA, and from there we get one protein. Sometimes there is alternate splicing, and so we can get more than one mRNA and hence more than one protein. But these just cannot explain the extreme diversity of antibodies that we have. In the 1960s, late 1950s, early 1960s, scientists estimated that the number of B cell receptors, which are basically what the antibodies are, must be somewhere around 10 to the power 7 or more, and they were all proteins. Now the question is, where is the space, or is the immune system itself having more than 1 million genes for 1 million proteins like these? This was very, very difficult to explain. In addition, as you know, that antibodies, immunoglobulins, are basically made of 4 polypeptides. There are 2 heavy chains and 2 light chains, and this is the arrangement, typical IgG arrangement. Now the light chains consist of around 214 amino acids. The heavy chains around 430 amino acids. Now what scientists noticed as they began studying these antibodies, they began to isolate antibodies and study them. What they noticed is that the N-terminal part of the light chains and the heavy chains, the first 110, 120 amino acids, was very, very variable. It varied from every single protein that they found. Very, very variable, and they may named it the V regions, the VL and the VH. The rest of the protein, in this case this part, in this case this part, this was more constant. Yeah, you could group them into 3-4 groups or 5 groups. Now the question is, how do you have a protein like this whose first half is so much variable, varying from every single molecule that they get, and the C-terminal part, the other half, the second half, is much less variable or overall constant, maybe 4 or 5 groups. Now how do you explain this? Now that would mean, given our conventional understanding, that you have genes whose one end has a lot of mutations happening and the other end has no mutations happening. Now how do you explain this? So this was the doubt, and there was no proper explanation for this. Then in 1976, Tonegawa and Hozumi, they did a brilliant experiment. In 1976, Tonegawa and Hozumi did a brilliant experiment. They basically took embryonic hepatocytes and adult antibody producing B cells. They isolated the genomic DNA from these hepatocytes and from the B cells, and they digested them separately with BAMH1. And they got DNA fragments, right? Also, from these antibody producing cells, they isolated light chain mRNA. Because these are antibody producing cells, they have light chain and heavy chain mRNA. And they labeled these with radioactivity. So, radio labeled light chain mRNA. All right. Now, so they had these fragmented DNA, and now they ran these on agarose gel. And what did they get? So they had these hepatocytes and B cells, and they ran these fragments on the agarose gel. And then they used that labeled light chain mRNA as a probe to see where were the genes which were binding, which were coding for this light chain mRNA. And they, what did they see? For the hepatocytes, they got two bands. So wherever the light chain mRNA was going and hybridizing to whichever DNA fragment, that would show up in the X-ray film as a dark band. So for the light chain, they got two bands. For the hepatocyte, they got two bands, and for the antibody producing B cells, they got one band. Genomic DNA, human genomic DNA, one from hepatocytes which don't produce antibody, one from B cells which produce antibody, same probe, light chain mRNA, radio labeled, you get two bands here, one band here. What is the explanation? The explanation is this. In the embryonic hepatocytes which do not produce any antibody, the variable region of the gene, there are the variable region and the constant regions of the light chains are coded not by entire genes but by gene fragments. So, for example, you have a variable region coding fragment over here, and that is separated by some non-coding DNA, and in distance you have the constant coding region. And because there is this gap, there was a BAMH1 site over here. It's genomic DNA, no? So you'll find BAMH1 sites in many places. So there was a BAMH1 site over here. And when they treated this, cut this DNA with BAMH1, you basically got two fragments. And when they probed it with the light chain mRNA, the light chain mRNA's 5' end could hybridize with the V part, and its 3' part could hybridize with the C part. So you are getting two bands. That is the explanation. So there is a gap between the V fragment and the C fragment. So, the light chains are not coded by one full gene, but by gene fragments. Now, what happens in the antibody producing cells? In the antibody producing B cells, this situation changes. There you get one band. Why? Because when B cells become mature and antibody producing, there is a genomic rearrangement and the V and the C regions come side by side. There is no gap between them anymore, and so that is why there is no BAM-H1 site in between them, because they're side by side, they're all joined. And now this light chain mRNA hybridizes only to this, and that is why you get one band, not two bands, because no BAM-H1 site is separating the V and the C fragments. This was very contrary to what traditional molecular biology would teach us. One gene, one protein, one RNA, all that. This was telling us that one polypeptide was being coded by one mRNA, true, but this mRNA was being formed only in B cells when separate gene fragments had rearranged their positions and had come side by side. This is something which is unique to the vertebrate immune system. It doesn't happen in any other tissue of the body. It doesn't, they experimented for embryonic cells. It doesn't happen in any other adult tissue except for the T lymphocytes, where you find a similar thing for the T receptor genes, T cell receptor genes. This subsequently was proved for all light chains, for all heavy chains, and now could explain why there were so many different kinds of antibodies whose variable region was varying so much. Let us see how. So now they began looking into these kind of sequences in detail, and they found that the heavy chains and the light chains were located, these gene fragments were located on different chromosomes, and yes, they were separated by non-coding DNA in non-Bs and B cells, but when B cells became mature, they came side by side. Now, the question is, you have so many variable gene fragments, right? How do you have so many variable gene fragments? And that is simple, because when they look for, first they look for the light chains, okay? First they look for the light chains, and they found that the first 97 amino acids, 1 to 97 amino acids of the variable region of the light chains is coded by a number of V fragments, and there are many V fragments sitting side by side with gaps between them in the genome. So you have something like V2, V1, V2, you get a gap Vn, so many of them sitting side by side. But if you remember the light chain, the variable region consists of 110 amino acids. Where is the rest? Downstream, at some distance, you have another fragment called the J fragment, and these are coding for the 98 to 110 amino acids. And further downstream, you have the C fragments. As such, they are separate in the genomes. But when B cells become mature in the bone marrow, then one of these V fragments combines with one of these J fragments, and they combine with the C fragment. And so you get a lot of combinations possible. Because let's assume there are 100 over here and 10 over here. So how many combinations are you getting? 1000. And that is why the variable region is coded not by one gene fragment but by two gene fragments, V and J. And that is why so much variation. There are multiple fragments like these sitting in the genome, and they combine randomly. And that is why we have so much a variation of the variable region of the light chains. How about the heavy chain? Similar, but bit more complex. The heavy chain's variable region is around 123 amino acids. And this, they found, is coded by many VH segments, VH1, VH2, okay, like this, VHN, whatever. So, and these code for the first 101 amino acids. And the J segment codes for the 107 to 123. And how about 102 to 106? There is another set of fragments called the D fragments, and they code for amino acids 102 to 106. By the way, D stands for diverse, J stands for joining. And that means the variable region of the heavy chains is coded not by two gene fragments but by three gene fragments. And when they join, you get something like one V randomly associates with one D, and they associate with the J. And finally, this associates with one of the C fragments, which is downstream. And there are at least five different C kind of fragments. And that is why we have 5 different kinds of antibodies: IgM, IgD, IgG, IgE, IgA. So the variable regions of the light chains are coded by 2 gene fragments, V and J, and the variable regions of the heavy chains are coded by 3 gene fragments, V, D, and J. Amazing, isn't it? And if we just look at how many V fragments and how many J fragments are there, the very high number itself tells us why so much of diversity of antibodies is possible. The best numbers are known from humans and mouse. I'll just show you the results for the humans. If we just plot them, so these are the heavy chains and these are the light chains. Now, I'm sure you know that light chains are of 2 types, lambda and kappa, based on their C-terminal constant region. Sorry, not C-terminal, constant regions. And let us see how many variable regions are there for the heavy chains, for the diverse regions, and the joining regions. This is 51 for humans. 27, 6, and any of these can associate with any of these, and these can associate with any of these. So how many combinations are possible? 8,262. For the lambda light chains, you have 31, no D here, and 4, that is 124. For the kappa, you have 40, 0, 5, 200. And these combinations are separate processes. Any of these, either this or these, can happen. So a total of 324 is possible, and these can combine with any of these. So what is the total value? What is the possible number we get from this? I'll give you the number right away. 2.7 into 10 to the power 6. By these kind of VDJ combinations, 2.7 into 10 to the power 6 different epitope binding antibody surfaces are possible, different variable regions, because it is the combination of the heavy chain variable region and the light chain variable region which will result in the epitope binding. And by these, potentially 2.7 into 10 to the power 6, 2.7 million different combinations are possible. And I assure you, we can discuss in another class, that this is further modified and the number of possible combinations even increases further. This explains actually why we have such a large number of antibodies which can bind to various pathogens, including a pathogen which is novel, like the new coronavirus, because the diversity already exists, and one of them will, at least one of them, actually more, will bind to one of the receptors, to the one of the proteins of the coronavirus. And that is the basis to our immune response, and that is what our entire vaccination system is banking on today. Fascinating, right? Generation of antibody diversity. Thank you.